everyone. Thanks for joining us. I'm Valerie Estes at Project ALS, along with my co-host, Erin Fleming. How's it going, Erin?
I'm doing well, Valerie. How about you?
I'm doing fine. We're here with an update for the community on the Phase 1b clinical trial of Prosetin, or P101. This is a proud day for me. Prosetin was first developed at the Project ALS Therapeutics Core over 10 years ago. Prosetin was designed to treat all forms of ALS. The core built Prosetin, improved its motor neuron rescue and medicinal properties. In other words, we studied Prosetin preclinically so that when Prosetin got to clinical trial, as it now has, it would have an evidence-backed chance of working in people with ALS. Project ALS believes in supporting research with a purpose. Our aim is to prove principle, but we do not make drugs. So, when Prosetin crushed preclinical milestones, Project ALS passed the baton of Prosetin to a newly formed biotech company. The goal of the new company was to deliver Prosetin to Phase 1b clinical trial in ALS. Investors in the company believed so deeply in the preclinical promise of Prosetin that they named the company Progenics after Project ALS founder Meredith's and my sister Jennifer. Yes, this is a proud day for me, Erin, because you've shifted your focus from Project ALS to Progenics and Prosetin. Now, you and your esteemed team will share what we know from the Phase 1b clinical trial in ALS patients. If you're watching this, we want to hear from you, so please send us your questions. If we don't have time to get to you, we'll find you afterward. Okay, Erin, let's get to the readout.
Thanks, Valerie. And just before we dive into the Prosetin update today with our stellar panelists, I just wanted to start by giving the lay of the land for today's discussion very briefly. Many of you already know that it was the Project ALS community, and probably, although I can't see you, some of you here today, that brought Prosetin to clinical handoff in the process that Valerie just described. In 2021, Project ALS, the Columbia Scientific founders, the scientists who invented Prosetin, and a venture capital group called Medical Excellence Capital started Progenics. And this is a biotech company, and the funding behind the biotech company and infrastructure has allowed us to move Prosetin from the preclinical stage at Project ALS into the first ever clinical trial of a MAP4K inhibitor for ALS. But I'm already getting ahead of myself because we haven't even described what a MAP4K inhibitor is or why we wanted to test Prosetin in ALS. And so to answer those questions, I'd like to introduce today's speakers. First, Gwen Peterson, who is the leading ALS advocate in the world and my heart since her diagnosis in 2018. She's a founding member of the Prosetin Community Advisory Board, both at Project ALS and now at Progenics. Dr. Jinsy Andrews, the director of the NYU Langone ALS Center, Neil's co-chair, also a clinical advisor to the Prosetin program, among many other hats and titles that it would take us the entire hour to get through. And Dr. Emily Lowry, an assistant professor at Columbia University, co-scientific director of the Project ALS Therapeutics Core, and Prosetin co-inventor. So we're going to share some slides without data throughout the next part of this discussion. But as Valerie said, before we start, I want to emphasize this is a discussion, not a formal presentation. If anything's unclear, tell us. We'll do our best to clarify and send us your questions again as they come in. We'll address them later in this webinar. All right, so Emily, let's start at the beginning. So how did you initially identify MAP4K as an ALS target, and what made you excited about this, in the big world of potential ALS therapeutic targets?
Yeah, we kind of went at this thinking, at that point, this was around 2016. And at that point, ALS drug development wasn't too different from where it is now, where there just weren't a whole lot of great options for people living with the disease. So our thought was sort of like, everything that's been to the clinic so far has failed. Instead of trying to, pick a target and go after it in sort of a narrow way, we decided to sort of screen really broadly and let the motor neuron biology guide us, and find drugs along the way, but also at the same time learn something about the disease. And really, actually, the story sort of starts even way before that. So like maybe 20, 25 years ago, when my boss, Henrik Victorly, was one of the first people with Project ALS funding along the way to be able to differentiate or to make a motor neuron in a dish. And once we were able to do that, we were able to build a cellular model of ALS in a dish. And that really allowed us to do the kind of drug screening that I was just mentioning, where we could just throw thousands of different compounds without really understanding what they were or what they did. We could just throw them on the cells, see if they rescued motor neuron degeneration in this dish. And what you're looking at in the upper left-hand panel is one of the first pieces of data that we pulled out of those screens. So each dot here is the drug. And then based on how high it goes, on the y-axis is basically how strongly it could protect motor neurons from stress. In this case, the stress that we were adding was a stress that causes protein misfolding that mimics what you see in people living with the disease. So you can see that there's a circle around protective kinase inhibitors. So one of those is one of the precursors that became Prosetin. Another one is kenpolone, which is circled. That's another one that we're working on now to make better versions of it. But that bigger circle that includes the precursor to Prosetin was called URMC99. It was just a tool compound. We didn't understand it. And the people who had originally developed it as a MAP3 kinase inhibitor. We don't need to get too much into the semantics, but basically it took us a few years to understand what the real target was. And we worked with some medicinal chemists to make better and better versions of the drug, and that did a better and better job rescuing motor neurons at really, really low concentrations. There's really beautiful work that was done as a collaboration for over 10 years with the Stockwell lab. And basically what you can see in the second panel on the top right, each dot there is a different version of Prosetin until we landed on the one that is Prosetin, which is in the tiny dot in blue, all the way at the top. So Prosetin is one of the most protective compounds we've ever seen in this ALS in a dish model. And it's a very, very strong inhibitor of a group of kinases called MAP4 kinases. Again, we didn't know what they hit. It took us years to figure out what they were actually hitting. So we ended up in sort of a champagne position where we had hundreds of drugs that worked and didn't know how they worked. So we spent a lot more time actually trying to unpack that. On the bottom, you can see what these screens look like in real life when we look under the microscope. So on the far left are healthy human motor neurons in a dish. And then in the middle, you see we've added a stress that causes all this protein toxicity, and the motor neurons disappear. The ones that are left, you can see a sort of round shape, which is the cell body, which is where all the genetic information, protein, and things like that live. And then the neurites, which are the things that would normally extend to your muscles, those are sort of all chopped off with stress. But once you add Prosetin, the cells look like they've never seen stress in the first place. And again, Prosetin is pretty much the best drug we've ever seen, hands down, in this kind of a model.
And I think you said you didn't know initially how Prosetin worked in ALS, and we're not going to spend, cover 10 years or 5 years of your life figuring this out here. But maybe just level setting around how we sort of figured out one key way that it was working before we move into the clinical update would be fantastic.
Yeah, so around the same time so actually, really, the reason that we thought that MAP4 kinases might be the real target, even though there are a bunch, the original version that wasn't very selective of the drug hit a bunch of different kinases. As we made it more selective, it looked like it was a better MAP4 kinase inhibitor. And around the same time, a bunch of other groups, this was like 2017, 2018, a bunch of other people were publishing that MAP4 kinases were super protective. But up to that point, everybody was focused on, people were focused on MAP3 kinases in neurodegeneration. It was something that I happened to study in grad school. That was the dogma, that MAP3 kinases lead to motor neuron degeneration. No one had ever heard of MAP4 kinases until, 2016, 2018. So it was really reading those other papers from other groups. Like we were really convinced that it was MAP3 kinases, but then reading those other papers, it was like, wait, we should maybe just check MAP4 kinases. And then it was like, oh, geez, like everything, they just totally lit up. So that's a very long way of saying we sort of came at it backwards, and then we figured out downstream these kinases that Prosetin can target sort of act in a linear pathway where a MAP4 kinase targets a MAP3 kinase, targets a MAP2 kinase, MAP kinase, and then so on and so forth until you get changes in gene expression that lead to cell death. So when you block these kinases upstream, you get a prevention of motor neuron death way, way downstream. But it also gave us the opportunity to sort of probe along these pathways to make sure that this is what we were actually hitting. And so what you're looking at in sort of the middle panel is a kind of readout called a Western blot. And this is just looking at the total levels of targets of the MAP4 kinases. And you can see under stress conditions, that target gets darker. Those bands get darker. That's an indication that the pathway is activated with stress. And then when you add a MAP4 kinase inhibitor like Prosetin or other selective compounds, that stress signal goes way, way, way down. And then really importantly, around the same time, other people were looking at the group at Genentech that were looking at tissues from post-mortem ALS samples. And they showed in the spinal cord that these same exact protein pathways that we were looking at go way up in tissues from people with ALS. So that told us that not only were we targeting this pathway very strongly with Prosetin, but that this pathway is sort of essential to the disease and to motor neuron degeneration.
Sorry, we have a good question from a viewer, Robert Foley. How long do neurons have to be exposed to Prosetin before the protective effects are measurable? Good question.
Yeah, the way that we do these screening assays, we actually add stress and Prosetin contemporaneously at the same time. But we've, everything is accelerated when you're looking in a dish, but we've pushed that. So we've added stress, and we wait, and then add Prosetin. And we can wait a few hours, which, it's hard to extrapolate that to a person, but we know that stress can already be underway. We can then add Prosetin after the stress has started and still stop it in its tracks. Hopefully that answers your question.
I would just add as the clinician that when you're looking, just like Dr. Lowry was saying, that she's looking at a model in a dish, this is an accelerated form of motor neuron degeneration. Yeah. So trying to take that information and applying it to people is a little tough. But we do know that we're able to rescue that motor neuron in that dish. So the hope is that we will learn as we develop Prosetin in clinical trials where the point will be where we see that benefit and the correlation or extrapolation of that information from the lab setting to the clinic.
And I think this is a perfect segue because we had all of this promising preclinical data, and now our next objective was really to see whether any of this could translate and whether Prosetin could be given to people. And as we thought about the clinical translation of Prosetin, one of the most important things for this program was forming a community advisory board. And Gwen, just to start, I'm hoping that you can sort of describe what your role has been and what the overall role of the community advisory board has been in the Prosetin development program.
Yes, happy to. Thanks, Erin. Again, I'm Gwen Peterson, living with ALS, and I've been a community advisor for Progenics pretty much from day one. And this map shows that Progenics was strategic in selecting advisors, people with lived experience for the board. They wanted to make sure there is an advisor in every country where the phase 1 study is currently underway. So thank you for that representation. Erin, I just want to chat about my experience in general as a patient advisor. So to date, I've been on more than 20 patient advisory boards, and I've got to say, ProGenX has been the most open, honest, and unfiltered board I've been a part of. And the reason for this is you guys have been really great about expectation setting up front with the advisor. So in in clinical trials, there are certain um, study design elements that are immovable, right? Like certain things that are baked in, certain things that FDA wants to see, has to see. And so, there's no room or little room to make changes. And really explaining a lot of the back end and what those either immovable or movable things are and why, and really like how we can be an asset as advisors is something we talked about from day one. Um, we also, as Progenex was submitting data and safety data and information with dosing, there were so many different FDA conversations and touch points that I know for me as an advocate, like I'm just not sure. I was never aware how the sausage gets made, like at that level. So really a transparency from Progenex around, the different touch points, deliverables, deadlines. It really helps me, one, like as someone interested in this process, understand, and two, kind of how to be a better advocate in general. As I mentioned, the Progenex board accurately reflects the ALS community geographically. It also reflects the advisors at different stages of our progression. It's really important to see, someone like me who can still speak verbally. We have others who communicate via eye gaze. We've got caregivers. We have advisors early in their journey versus several years in. So it's definitely a range of perspectives. And yeah, I think, Erin, is there any other questions? Anything else I should cover?
No, I I think you've said everything that's important to me about this board also. And I think, of course, Gwen, just chime in with anything else that has informed your
Yeah.
experience along the way. But why don't we get into the clinical trial? And Jinsey, I'll start with the study design. Maybe you could walk through this, but also maybe even before that, just from a high level. And I know this is a very basic question to ask, but what is the purpose of a phase 1 study in ALS? Like, what are the non-negotiable things you want to learn? And then what are you trying to also learn potentially in a phase 1 ALS study?
I think this is like a super important question, and I'll tell you why. It's because it really pertains to the mechanism of kinase inhibition, and the work that Project ALS and Progenics has done over the course of the last several years to really refine the drug before it actually enters clinical trial. Kinases, just for people who are not familiar with it, is essentially a set of enzymes that are like on-off switches in our body. And kinases work all over your body. So some important points that Dr. Lowry pointed out was really critical before entering this phase one. One is that, why do we select MAP4K? It's very selective. It was effective in preventing motor neuron degeneration in the lab setting. So we really had some strong confidence that that target was really relevant and a high potential for it to be effective in treating ALS and maybe even answering some of the questions you guys are building in the chat there. So phase one becomes relevant for not just kinase inhibition, for all drugs, whether that's targeting mitochondrial dysfunction or nerve inflammation or even stem cells or T cells or whatever it may be that we're using in ALS. It's really critical to do two things in phase one. One is to really assess the safety and tolerability across a range of doses and hopefully get to the dose that we believe to be the most therapeutic dose for ALS. The second is to optimize the dose, right? So we have to understand safety and we have to understand the optimal dose that we want to carry forward. And why I'm talking and emphasizing those 2 points is in drug development, and I've been doing clinical trials for now over 25 years, this is the part that a lot of people skip. And in the urgency and the haste to move forward into a phase 2 or phase 3 study, we don't really understand the drug itself, the mechanism. We don't understand the true safety potential. And I've worked on drugs that looked good preclinically, but we didn't really understand the safety and jumped right in. And something like, I'll use an old example, minocycline. It's the drug that people use for acne and it's thought to be very benign. We moved right to a phase 3 study that was NIH funded, and it had a whole slew of side effects that were unexpected and not really seen often because it's used in short duration in other populations. And it also caused faster decline in people living with ALS compared to placebo. So it's really important, even if the drug's been around for a long time, to understand safety and tolerability and understand the mechanism of the drug. So that's why phase one and this phase is critically important. Gwen?
Jancy, so it's possible to skip phase one altogether? Skip over yeah.
So the for new chemical compounds that have never been used in another indication, you have to do a phase one. But if there are drugs or components of a drug that are already FDA approved, sometimes people will just jump right to a phase 2 and a phase 3. I don't have to list them all out, but if you look, and now ChatGPT's out there, you can look at all the drugs that have been tried and failed. A lot of them are repurposed drugs.
Right.
And often we might have missed something, or even supplements. We might have missed a signal, that might have been a different dose or might have carried a different safety at a different level. So that's why this phase one
Thank
portion
you.
of safety and dose optimization is important.
Again, it bears repeating, but Prosetin was built from the ground up for the treatment of all forms of ALS.
And that really informed how we designed the study because we needed to test different doses. So the part C, the first part of the study, was a multiple ascending dose portion, which is exactly what it sounds like. You test different doses starting at low doses, and then once you understand the safety of each higher dose, you can continue until you find a dose that is hopefully at the range where you would expect to see benefits and is also not causing safety effects. And that is for a new compound like Prosetin, and I think, as Jinsey is saying, really for all compounds, a really critical step, whether it's a repurposed drug or not. But in this context, certainly we needed to understand if there was a dose of Prosetin that was both safe and active that we could move forward. But of course, we also didn't want to stop there, and we wanted to have the opportunity for long-term treatment and also learn more because with only short-term treatment, we really won't learn anything about what Prosetin might be doing longer term in ALS from a safety perspective or from an activity perspective. So there is an open label extension that's now 2 years long that's open to all participants that go through the dose escalation portion.
Hey, the
Sorry, Jinsey.
yeah, the I'm part of the community, so I'm going to ask a question. Everyone's looking at part C and part D. What happened to part A and B?
So we omitted those from this webinar for time's sake because these parts, because it was a brand new drug, and this actually came out of our conversations with FDA before we started this clinical trial. We had what's called a pre-IND meeting where we went to FDA. Jinsey helped in designing a study, and we discussed the plans with FDA. And they were very clear that because this is a new drug and because it's actually a new mechanism. There's never before been a brain penetrant kinase inhibitor that's been tested in people. And so FDA said you have to follow a very standard study design, which included, even before all of this, a single ascending dose portion where each person just took one dose of Prosetin and saw what happened after. And so we ran that part of the study and looked at some lower doses in short-term dosing in healthy volunteers because it was just much faster to do it that way. Anyone who enrolls in the single dose portion can't enroll in the later part of the study. That's another regulatory rule. So it really was not, there was no benefit to trying to run that part of the study in anything other than healthy volunteers because there was no chance of having any benefit from the drug.
And I asked you that question on purpose because I will say one of the unique qualities of this clinical development program is that we used what Gwen described, the community board. So the value of using an investigational product or an experimental compound, giving only one dose doesn't mean anything to people living with ALS. They want to have access, should they choose that as an option for them, and have it as long as possible, as long as it's safe and tolerable. So we really tried to meet the needs of people living with ALS and took the advice of the community board, tried to mix that or blend that in with the advice given to us from the regulatory perspective, and really successfully developed a program that could move through and meet all of those needs and requirements. And the open label extension is part of that discussion, going back and forth with the community board and allowing extended access to that investigational product as we learn about it.
All right, so let's get into some data. I'm going to skip a slide about how safety was assessed because I think, Jinsey, you can talk through that without a slide. But really, what have we learned about Prosetin safety in PRO 101? And Dr. Andrews, I'm going to hand that over to you to start to guide us.
Yeah, so I mean, the big picture is, as I mentioned before, phase one, the purpose of it is really to understand safety. As you learned from Aaron, this was a new chemical entity, it was a new target, and so we really wanted to be sure that we're bringing something that has high potential for benefit in ALS and good scientific rationale and backing, as well as understanding its safety and optimized dose. So what we learned through the process of doing this phase one study is that there were no serious adverse events that were reported to date with PRO 101. We also really didn't see any significant trends that were observed during the study safety assessments, and we did very rigorous safety assessments. And then at the higher doses, some participants did develop some oral irritation. These were really concentrated liquid dosing, and they resolved once the drug was stopped, and so they were considered not serious. And of course, some of this information helps us optimize the formulation and the dose selection. So we would consider figuring out the ideal formulation in future studies going forward. But essentially, the big picture here is that it was very well tolerated and it was very safe, and it gives us good confidence to take it forward in a further development for Prosetin.
And I do see an audience question that's
Yeah.
relevant here. From what I see about the trial, you're administering the Prosetin in liquid form, and it will cross the blood-brain barrier. Was there any reason not to administer it in pill form or intrathecal? And really, we purposely developed the liquid formulation of Prosetin. We wanted to keep in mind swallowing considerations, also feeding tube considerations, and have a formulation that would be easily adaptable across the ALS spectrum. I think that because we have seen oral irritation, we have a small dose. And so I think that it's likely that we might be able to address this by just changing the liquid formulation, which would be our preference. But a pill form is certainly on the table. That's something that would be very possible to do if we needed to move to that in a future clinical study. And in that case, we would just need to make sure that again, we have compatibility for feeding tube administration and sort of considering across the spectrum as we need to adapt the formulation. So hopefully liquid will continue. If not, a pill is certainly something that would be not that challenging. I mean, It is challenging. Manufacturing is always challenging, but certainly possible to consider.
Yeah.
I just want just a program note. We're going to get to certain questions a little bit later after Erin completes her presentation of the data. But these are all great questions, and thank you. Keep them coming. Take it away.
Okay.
And Erin, really quick, because phase one passed the safety and met the safety and tolerability standards, the board has fully backed sort of pushing into phase two. So yes,
Thank
we
you.
support a phase two study.
Yes, and we'll talk about next steps very soon, but that's an important mark of confidence for us as we think about what's to
Yeah.
come. Okay, and the other really key endpoint in this study was pharmacokinetics, and maybe Dr. Andrews, you could describe what that means.
Yeah, I think I'll just highlight that when you're giving drug to people, and this goes back to the question of giving it orally versus giving it in the spinal fluid versus giving it in the blood, we want to make sure the drug gets to where it's going and we get to the levels that we want to. So that's pharmacokinetics, understanding how much drug gets into your body and whether it gets to the level of where we are targeting a potential therapeutic effect. So we were able to see in the shaded area, and Erin, you can explain a little bit more about the targeted therapeutic levels that we were trying to aim for. But we really took the time to understand the dose that was given in ALS and measure the actual concentrations in each of those cohorts as we increase the dose levels so that we reached the ideal or optimal therapeutic range. And then also look at the safety in combination to see where the sweet spot would be to carry it into a phase 2 study.
Yep, exactly. And if anyone has specific questions about where this blue bar came from, I can answer them here or any other time in more detail than you'd probably like. But essentially, this came out of a preclinical model that we used that we tested Prosetin at different doses in. So we could see sort of the optimal dose for optimal neuroprotection in an animal model. And then we used that. We looked at the drug levels in those animals and used that to set this range here for people. So again, in a phase one study, you're always extrapolating from what's happening in a cell or what's happening in an animal to what we hope will happen in a person. And this is the grounds for us to evaluate it. But we did have a rationale for setting that as our target therapeutic range. Um, and the other really critical thing that we focus on in the study is trying to assess the actual target engagement of Prosetin. And Emily, I'm going to ask you to talk about this first because you spent many years of your life working on how we could actually measure whether Prosetin was hitting MAP4 kinase in a person. And I know we don't have
Yeah.
time to, again, get into great detail, but it would be really helpful for you to just explain why this was so important to us and sort of what your approach was.
Emily, can you quickly define target engagement?
Yeah, I was just going to say, thanks, Gwen. Yeah, so Dr. Andrews was talking about pharmacokinetics, and the kinetics means how a drug moves through your body. So that we nailed down, we could see Prosetin where it needed to be. But pharmacodynamics is the other sort of half of that, which is if the drug is where it needs to be, is it doing what it needs to be doing? Is it hitting its target? So Gwen, that's, yeah, another term for that is target engagement. It's, literally like a lock and a key. Prosetin is hitting what it needs to hit, these MAP4 kinases. So as Erin mentioned, this was really tough thing to figure out. We had designed Prosetin to be very brain penetrant, and basically it was very, very good at that. So in our preclinical models, when we were looking for a biomarker of target engagement, we had a lot of trouble because basically, Prosetin goes straight into the brain. It doesn't spend a lot of time in your bloodstream, but really the only accessible or the most accessible tissues that we could get to test whether Prosetin was doing what it needed to do in a patient would be in the periphery, in the blood, not in the brain or the spinal cord. That's very hard to access. So we needed to look for sort of a proxy of Prosetin activity that would mirror what it's doing in the brain and the spinal cord, but something that we could sample from the blood. And so we threw a lot at this. And ultimately, the thing that ended up working the best was the thing that was the most logical. And that almost it was one of the first things that we started with. And in the middle panel, you can see again, this sort of linear pathway, this enzymatic pathway that Jinsy described, where MAP4 kinases target MAP3 kinases and so on. And in the middle of that pathway is MAP2 kinases. And what you're looking at on the left are motor neurons that have been stressed and or treated with Prosetin. You can see that Prosetin decreases the intensity of that MAP2K signal, that stress signal. So basically, we wanted to see, can we see this decrease in the stress signal that we know is downstream of MAP4K? Can we see that in the blood? And again, it took a lot of time and refinement and working with a really lovely group of researchers in Sweden to ultimately make it work. But on the top panel on the right, you're seeing Prosetin in blood from animals that have been treated with Prosetin. And then one of the most exciting days, I think, of this whole process was seeing the same result in the blood from people who had been treated with Prosetin. So you can see that stress signal, that MAP2K signal, going way down when you add the drug.
And one of the things that we saw, so just looking at this in more detail, this is the actual data. So each of these dots is a blood sample collected from a person in PRO 101. And we collected them before anyone received any drug, and then after they had received Prosetin for 14 days. And this is a complicated slide, and this is very new data, so we actually have only looked at it, in very basic ways so far, and have a lot more to learn about this pathway and this target engagement marker. But I think what you can see very clearly is for people who received placebo for the first 14 days, there's no change in what's happening in this pathway. For people who received lower doses of Prosetin, so these are the concentration, the drug levels of Prosetin that are a little below where we would like them to be and we think is needed for activity, ideal activity. You see a slight decrease, but it's just modest. And then in people who received higher doses of Prosetin and had higher concentrations in their blood, you can see a much bigger decrease in the activity of the pathway. So this is data that we're going to be spending a lot of time with, but really this was critical for us because, as Emily said, we could look at safety, and that's great. Having a safe dose to move forward is perfect. But if we don't know whether that's active or whether it's actually doing what we think it should be doing, inhibiting MAP4 kinase in an actual human being, then we really don't know whether that dose of a drug has any chance of actually having an impact on ALS. And I think, as Dr. Andrews referred to earlier, there have been so many drugs where there's a late stage clinical trial that doesn't work. And at the end of the day, we don't know whether it's because the target was wrong, is MAP4K the right target? We don't know whether it's because the dose was wrong or if there was just something else we were missing in the entire process. So maybe I'll stop sharing.
No, if I could just actually jump in there, because
Oh.
Nadia Sethi asked an excellent and related question. Why did Progenics take this approach? Like, why have we front-loaded the Prosetin program? Like, how do we know so much about Prosetin? We're only through phase 1. I mean, I think it's a great thing, but why don't more people do it? Why don't more
Dr.
companies
Andrews,
do
do you
it?
want to take a first pass at that?
Yes, I mean, having been in the industry setting where I'm developing a drug, I definitely see where things can be efficient. And one of the areas I think in ALS, we've struggled with trying to get game-changing drugs out there, especially for sporadic forms of ALS. And so one of the weaknesses is in this phase of development. We don't spend enough time here because the investment of time and resources in this area, you get the return on that investment later when you're developing a drug in clinical trials, and you have more confidence in answering questions about how long somebody should be on it, who are the right people, what are the markers that we need to look at. So that's kind of why I think we've spent so much time in this early phase preclinical and early phase clinical development. It is probably one of the things that makes this so rigorous and thoughtful in terms of Project ALS's approach and Progenics's approach, and also communicating it out and explaining why we're doing this in this manner, because it's really important for us as a community to understand if we really want drugs to work, we have to do the work here.
Nadia, I hope that answers your question.
I just
Yeah, go.
wanted to quickly, I see a couple of questions that we missed
Yes.
during the conversation, so why don't
No, they
we
weren't.
see what we
I
can
wanted to
do?
hold them until we had this clinical conversation and next steps conversation.
Yes.
But please answer them, Erin.
So quickly, just Robert Berman, I'm sorry we missed your question, but yes, Prosetin is brain penetrant. We've seen this across all of our preclinical models. We're going to be, throughout the PRO 101 study, we have included lumbar punctures. And we did that because there's very valuable data. I think for a CNS penetrant drug, we really want to be sure that we're seeing the same type of CNS penetrance, that it's actually getting across the blood-brain barrier in people as well as it did in animals, as another important data point. So we'll be looking at that data very soon. But in all of our preclinical studies, we saw that it did cross the blood-brain barrier. And then
Aaron, question on that from me. Shouldn't all drugs for neurologic, neurodegenerative diseases be brain penetrant and cross the blood-brain barrier?
I think the obvious
I'm-
question to that is yes, but are they is a different question.
Right.
And I
Right.
defer to our experts on that.
Yeah.
Right.
And I will say it also depends on the target. So if you're trying to target motor neuron degeneration and degeneration that occurs from the brain and the spinal cord, then you really need to be brain penetrant and you need to cross the blood-brain barrier. There are some novel strategies like approaching the neuromuscular junction, or where the nerve and muscle meet, or in the muscle, which is where I was doing work for a little bit. And we didn't want it to cross the blood-brain barrier, but it did because we didn't study it. And so those, it really depends on the target. And this is why it's really important to understand how a drug behaves in your body. So whether it crosses the blood-brain barrier and whether it's brain penetrant, but also understanding once you get it there, is it doing its job? That's target engagement.
And
Yeah,
that's what we have.
I was just going to add that the Prosetin is kind of a unicorn, and we worked really hard with the chemists who are collaborators on this project to make it so brain penetrant. It's very rare, especially for kinase inhibitors, for them to be able to penetrate the brain, because your body has natural ways to try to flush drugs out, the same mechanisms it uses to keep out bacteria and viruses. It really doesn't want outside things in your brain. So we were able to sort of override that with some chemical tricks, but that took years also to make sure that was working.
Thank you. We have a couple other questions. Mark Friedman, thank you for your patience, first of all. But he asks, if Prosetin works, do you know, or what is your guess of, and what stage will it be effective? Will it work for someone with symptom onset under 1 year, 2 years, 3 years? Or will it be effective if your neurofilament light chain level is below a certain amount? Thank you.
I guess I will take that one first, unless Erin, you want to jump in. That's a tough
Please.
question.
Yes.
And I will say that we don't have definitive answers quite yet, but we've tried to leverage, even in the first phase of development, trying to give prolonged exposure in the open label extension and understand how that affects ALS, but it's not going to be the totality of the information that we need to answer that particular question. Theoretically, and Dr. Lowry, please correct me if I'm wrong, theoretically in ALS, there are things that are occurring in the motor nerve, like oxidative stress and abnormal protein aggregation. And these things kind of trigger stress. And so when it does that, it kind of activates, it's a signal that activates this MAP4 kinase, and the switch is turned on, and that leads to motor neuron death, as you saw on the slide earlier. So if you think about it, when these things are occurring, it almost seems like you want to catch it a little bit earlier in the stage of disease rather than later when the motor neurons are already not there. So theoretically, if you think about it scientifically, the hypothesis would be, we would want to try to protect those nerves and prevent them from dying. So the earlier, maybe the better.
But does
Yeah,
that mean, I guess just a follow on to that, Jency, is we can still expect efficacy if we're farther along, because what we've seen in preclinical work is that Prosetin rescues motor neurons when they're super stressed.
Yeah, and when they're sick, the beauty about the phase one is that we were very flexible about the inclusion exclusion criteria, and so we didn't set a time of symptom onset or disease duration. We just required some minimum respiratory function, the diagnosis of ALS, that people be adults, and we included them in the study. So we were very broad on the eligibility criteria for people to participate in this early phase. So we had some experience with people who are a little bit further along than something like that we traditionally do in later phase development.
Yep.
Great.
I
And
think
thanks
to
for the
that,
question, Mark. Yeah.
I just wanted to add some specifics because we did purposely, for a lot of reasons, have very broad eligibility criteria in this phase one study. And the data that we haven't presented because it's still ongoing as the open label extension is ongoing are things like we've looked at the baseline neurofilament light levels for every participant in the study. So we will look at whether there's any difference in, inflammatory pathways based on some biomarkers that we're looking at and time since symptom onset, site of symptom onset. We'll be able to look at whether there's any variability in how the outcomes in the study track with those baseline characteristics. The very important caveat is that this is a small phase one study. And so we're not going to know definitively, even when we look at data from this study, like, yes, a neurofilament light chain level of X is what we need, to respond to Prosetin. That's not a question that the study is equipped to answer. But we tried to include as many outcomes, functional and biomarker outcomes, and this very long-term dosing so that we can at least gain valuable data that will help us to design a phase two study in the right people with the right endpoints in a scientifically thoughtful way.
So just to be clear, Aaron, there's a further readout to phase one. There's more data to come and to share with the community. We might want to think of reconvening later this summer to read out the rest of it. There's a great question here from Paula Roxana Katana. And thank you so much. I think it has Dr. Emily Lowry written all over it. Is the MAP4K pathway inhibition expected to stop immediately after discontinuation or decline gradually? What about its neuroprotective effects?
Yeah, I was just going to say, actually, this dovetails nicely on what I was going to say earlier, which is that Prosetin was designed to target these kinds of cellular stress that are common to all forms of ALS. So motor neurons, across the board would be experiencing this, and it's sort of a gradual process. It's not all at once. We don't totally know exactly what's happening in patients, but we know that that motor neurons can be sort of protected. The idea would be that Prosetin would sort of pause these stress signals and give motor neurons a chance to sort of reset, clear all of this misfolded protein and all this debris, and try to basically the surviving motor neurons that are there, give them a chance to take over for the motor neurons that may have already been lost. Timing wise, it's difficult to say, but I think that we'd have a good chance of if there are motor neurons still there and surviving, of keeping those guys resilient.
Let's keep those
really
guys
helpful.
resilient.
All right. Well, any other questions from the community that we can answer now? I think we've gotten to everything, unless my eyes deceive me, which they sometimes do. Progenics team, I want to thank you so much for this excellent update. We're quite excited at Project ALS to see what comes next. We're super jacked to sorry. We're really excited to learn more about what the phase one readout means or says. Maybe it doesn't mean that much, Aaron, as you said, because a phase one study is typically smaller, but still target engagement. It's unusual to have this kind of data at a phase one. we at Project ALS, and on behalf of the ALS community, just want to say we're so proud of Progenics. Thank you for all you're doing every single day to get Prosetin over the line, and we're really excited to see what comes next.
Thank you, Valerie. Thank
Oh,
you.
and can I also apologies to Dr. Jinsey Andrews for the misspelling of her name. That is Dr. Jinsey Andrews with an S, Dr. Emily Lowry, Gwen Peterson, Aaron Fleming. Thank you so much. And I do want to shout out to Progenics and its leadership, which has been so generous and so committed to the furtherance of excellent ALS research, and in this case, Prosetin. They really care, and I think that's part of the big difference between this and other programs.
Yeah,
So thanks, everyone.
congratulations. No.
Yeah.
Thanks, Valerie. Thank you, everyone.
We'll talk to you guys
for joining.
soon.
We'll
Thank
talk to
you.
soon. Have a good evening.
Bye-bye.